Okay, welcome to Microbial Growth. Um, we've got to be able to grow these organisms out in order to identify them. So there are certain things, until we could figure out how to grow them out, we could never see them except underneath the microscope where they're little tiny little things that we stain. Okay, so in order to grow these out, we have to provide certain things for these microbes to grow. Now, first of all, you have to understand, unlike humans and animals, when we grow in height and weight. We don't grow in numbers. Microbial growth means we increase the population. We make colonies. We go from one colony to a million colonies. So microbial growth in these bacteria is an increase in the population number, which is different. And this is a result of these discrete colonies replicating by binary fission, as we talked about. And so each colony that you see on a plate is a bunch of cells, an aggregation of cells coming from one single parent cell. And they can be millions of cells in that colony. But they're all the same. They all have the same genetics because they've been doing binary fission, and they all have the single parent, and that parent has reproduced a million times. So it's just in growth in numbers, not getting, bigger in size. These organisms need a variety of nutrients, and we have to provide them. And some of them can be a little hard to deal with, and some of them are very easy to grow. So we have some very fastidious organisms where we have to provide them specific environments or they don't grow, and some grow very well. All right, so the most common nutrients that we need is CHON. That's how you remember it, CHON. Carbon, hydrogen, oxygen, nitrogen. All living organisms require this because you have to be able to breathe and you've got to make proteins and that sort of thing. And that's all these elements are in that. Sorry for the noise. I just decides to work out in the yard. My neighbor. And so these microbes obtain their nutrients from a variety of sources, and we're going to talk about, we classify them according to that. So we have chemical requirements and energy requirements, and there's a group of organisms based on their how they get their carbon, because we said they need carbon. Some of them use inorganic carbon, i.e. carbon dioxide. Others get the carbon by, breaking down organic molecules and obtaining them. Okay, we'll go over this in a second. Then you have two groups of organisms based on whether they use chemicals or light as a source of energy. If you're phototroph, you use light. If you're a chemotroph, you have to get it from chemical reactions. So let's look at this chart. It's pretty good. All right, so we have we put the photo together with either the auto or the hetero, so it goes together. So we either have photoautotrophs, chemoautotrophs, photoheterotrophs, and so on and on. Back. All right, so photoautotrophs, as we said, let's go back to our definition of auto. Okay, they use carbon dioxide as an inorganic source. Okay, so these guys use light. So these are plants and algae and your cyanobacteria, blue-green algae. These bacteria use water to reduce carbon dioxide, and they produce oxygen as a byproduct. Your photosynthetic bacteria, there's a couple of them, this green sulfur bacteria and the purple sulfur bacteria. They don't use water, nor do they produce oxygen, but they can photosynthesize. Okay. All right, so we'll go across the chemical compound, the chemoautotrophs. These are a very kind of narrow group of bacteria. They use hydrogen or sulfur or nitrifying bacteria. Nitrifying bacteria are very, very, very important, just FYI. They allow plants to grow. They form nodules on the roots of plants, and they convert this nitrogen so that the plants can use them. So nitrifying bacteria are very, very important. Then we get down to those organisms that use organic compounds. And again, first of all, you can have the photoheterotrophs, which means they do photosynthesize. But the big, 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 big group, the main group that we're going to be working with is chemoheterotrophs. These guys are us, okay? We use aerobic respiration. We cannot photosynthesize. So by definition, we eat. Whatever we eat, we break down. We breathe in oxygen. We break down these chemicals to whatever we need. So our carbon source is these organic compounds that we break down. So this is going to be most of the organisms, any organism that does aerobic respiration, some of these bacteria that actually do anaerobic respiration. And then we'll talk about fermentation. Fermentation, basically, converting some of this. We'll talk about what it is, but bacteria and yeast can do this. Okay. Those of you that have had some chemistry have an idea what fermentation is, but it's that conversion. All right, so let's look at these requirements. First of all, oxygen. Obligate. We have some definitions of these things that help us. Obligate aerobe means it has to have air to live. There are organisms that don't have to have air, oxygen. But obligate aerobes means they're obliged or required to have oxygen. Okay. So they are doing that electron transport chain and they need that oxygen. All right. If you have an anaerobic bacteria, oxygen is deadly for them. And we have to make special provisions in the laboratory. We have to pump CO2 gas into the incubators to keep the oxygen low so that these guys can grow. And we'll talk about this when we talk in the lab about transporting these organisms. You have to realize that for these anaerobes, certain forms of oxygen are highly reactive and they're toxic for them, okay? And it can cause irreparable damage to those cells. So if you have an anaerobe, you absolutely have to provide a non-oxygen environment or very low, okay? And what we do is we pump in CO2. All right? Okay, so there are 4 toxic forms of oxygen. Singlet oxygen, most oxygen is found as O2. This would be an oxygen radical. All right, and this occurs during photosynthesis. So your phototrophic organisms have these carotenoids. You ever heard of this? This is the protein is a coloring agent that gives their color to like carrots, stuff like that. That particular compound removes the excess energy of the singlet oxygen. So not really biggie. They'll be the one I really want you to know. Superoxide radicals, they form when you don't reduce the oxygen completely in aerobic and anaerobic respiration. So they do react to reduce these superoxide dimutases and to detoxify them. Anaerobics don't have that enzyme, and that's a lot of times why they die. We have that enzyme to detoxify these radicals. Anaerobes don't, and they die as a result of this. Okay, but that's all you remember. I'm fine with it. Peroxide ions can form from this enzyme. Anaerobes contain either catalase or peroxidase, these two enzymes that detoxify peroxide, so not a problem. Obligate anaerobes either lack both of them or only have one of them. So again, that will kill them. So if you're an anaerobe, you got a problem. Get anywhere, get near oxygen. The most reactive form of oxygen is this fourth one, which is hydroxyl radical OH with a negative sign. All right, and it results from ionizing radiation. We'll talk about killing organisms using radiation, or the incomplete reduction of hydrogen peroxide. It is very reactive. It is the most toxic form. It's not a threat to aerobes because of our catalase and our peroxidase. So, basically, if you're an aerobe, you're fine. You don't care about this. If you're an anaerobe, you got a problem. All right, that's the bottom line. Okay, so we classify these guys by their oxygen requirements. Aerobes undergo aerobic respiration. We like to be in air. We have to be in air. Anaerobes, they don't use aerobic metabolism. So they're not going to really want to be in a lot of oxygen. Now we have some that can tolerate it for a while. Facultative anaerobes can maintain life via fermentation or anaerobic respiration or by aerobic. So they can tolerate it. They can work in that area, but they don't like a high oxygen content for very long. Aerotolerant anaerobes do not use aerobic metabolism, but they have some enzymes that detoxify the forms we just talked about. And then these microaerophils, these are aerobes that require oxygen levels from 2 to 10%, and they have a very limited ability to detoxify. So you got to keep them in that low oxygen environment. So if you put them in a tube, this is what it looks like. Okay, and we can do this in the lab. We would have done this, but since you're not a lab, we can't. But you can see this oxygen concentration. You take a tube of broth. You got to take the top off because some oxygen gets in this tube. You put your organism in there and you close the tube up. And always when we do this, when we use broths, we have to back that top back, because if you have an aerobic organism, you got to get oxygen into this tube. Okay, so obligate aerobes are always going to grow up here at the top where the oxygen is, because the oxygen has come in, is leaking in, and the oxygen's here, and so you're finding oxygen here. So if you see the organisms only grow here, you've got an obligate aerobe. You got an organism that has to work with oxygen. If everything's down on the bottom, then you've got an obligate anaerobe, because this is now, as you go down the tube, the oxygen level goes down. Okay. Facultative anaerobes, again, they can work with that oxygen. You can see them as they scatter down, whereas aerotolerant does not tolerate as much oxygen in there, so they're going to grow throughout. But you can see these two. All right. Nitrogen. We have to have nitrogen in order to make proteins and nucleotides. Nitrogen is acquired by breaking our nutrients up, and we recycle those nutrients into amino acids and nucleotides. You can reduce this nitrogen to ammonia by certain bacteria, which is essential. As I mentioned, these nitrogen fixing bacteria take this nitrogen gas and reduce it so that these plants make the nitrogen available to these plants in a usable form. Okay. Other things that we need is we need phosphorus, because that's part of our nucleic acid, ATP, think about the name, what ATP means. Sulfur is required. A lot of things require disulfide bonds. So you need that in amino acids and that sort of thing, and certain vitamins. And we have trace elements that we usually get that are in the atmosphere, in the environment, and certain growth factors that can't be synthesized, like we take amino acids and vitamins and stuff. So there's other chemicals that are needed, and we put all this stuff in. Okay, when we try and grow these bacteria, it's not necessary that all that, but we take that stuff and we put it in, and it is in either the broth or the agar where we're trying to grow these bacteria. If we don't provide the certain amino acid that the organism needs, the organism can't grow. Okay. I've seen instances, and we'll talk about it later, but if you've got, a new disease coming out or something like that, where you don't know what it is and you're trying to grow these organisms out, and we've had these cases, and it's just you can't until you keep experimenting with amino acids and nutrients until you can grow these in different temperatures and different oxygen levels until you can actually grow these guys out. Okay, it does happen. It's happened plenty of times, and it's just you've got an organism. You can see it on a slide, but you can't grow it out. If you can't grow it out, you're missing something in your requirements that organism needs. But most of the time we do very, very well. But there are certain, I will tell you, when we actually culture organisms, when we send a swab down to the lab and we culture them out, we don't just put them on one plate. We don't just put them in one broth. We will culture them out on several different plates and put them in different temperatures and different oxygen levels to make sure, depending on where the specimen is coming from, to make sure we try and get all these organisms to grow. Okay, just FYI. All right, temperature, big issue here. Has a huge effect on proteins. Hopefully you know that. You can denature protein by getting too high, or you can stiffen these proteins up so that everything becomes too rigid and they fail to grow. So has a huge effect on lipids, has a huge effect on lipid-containing membranes. We have our membranes around their cells. So again, if it's too low, the membranes become very rigid and fragile, and too high, the membranes are too fluid, can't contain the cell, and proteins denature. Okay, so if we look at a temperature curve, each of these organisms have optimal temperatures. Okay. And we do classify them. I'll show you this in a second. So what you do is you're looking for this optimum temperature, whatever it may be. Now, this particular organism, you can tell, likes 37 degrees. All right, 37 degrees is body temperature, 98.6. All right, 30 is getting a little low, but it's doing fine. But 22, which is basically around room temp, 22, 25, you don't like room temp. So if I try and grow this out at just room temperature, it's not going to grow very well. This is why when you go into a microbiology lab, we have a incubator that is set at 37 degrees. We have an incubator that's set at a little bit cooler temperature. One is at a little warmer temperature. So we set these again at different temperatures to make sure that we're going to hit all the optimum temperatures for these organisms. So for the one that's at a 37, that's optimum 37 temperature, you can see you have minimum growth at the cold, begins to grow. When it gets up in here, it hits that optimum temperature, but it very quickly goes down because then it gets too hot and those proteins denature. So we do classify these organisms by the range of temperature growth. All right. So if we like it cold, and there are some organisms that like it cold, and we have to be careful with them because they like refrigerator temperatures, and this is why you can get growth in your refrigerator. All right, this is Celsius, so this is what? Think about it, 0 degrees Celsius is what? Yeah, freezing. So you've got, here's 37, all right? So your cold guys that like it just above freezing or slightly below freezing, you can grow organisms in the freezer. All right, they're psychrophiles. Then you guys have the guys that like the middle temperatures. These are the guys that like body temperature, our body temperature, made for our body temperature. So these are the mesophils. They like it right around 30, 37, and you can see they're really happy there. Once it gets up to 45 degrees Celsius, they are dead. They're not happy. Then we have those that like it warmer, thermophils. And then we have those that really like it hot. Okay, we're kind of going to look at those. But these are the guys like in the volcanic vents or in the hot springs. They like it hot. Okay, they like it hot. Then we have pH requirements. All right, organisms can be very sensitive to acidity. Some of them like acids, some of them like bases. Most of them like it neutral. They like a pH between 6.5, 7.5, somewhere in there. That's most of us. So most of the bacteria in the protozoas grow best in that normal range. Gee, that's our normal range. These guys are adapted to us. The acidophils, they grow best in acidic environments. So, you can use them to clean up waste products, and you can help use, they help preserve foods by preventing microbial growth in pickling and that sort of thing. Guys that like the bases, they live in these alkaline soils and water up to 11.5. That's very alkaline. So you can get guys that can grow in these really extremes. Okay. All right. Then we need water. All right. Microbes do require water. We have to have water. Most cells die in the absence of water. Not all of them do. Some have cell walls that retain the water. We'll talk about endospores. Endospores form and they just cease the metabolic activity in a dry environment, or can be for years or decades. We'll talk about endospores. But there's two physical effects of water: the osmotic pressure and hydrostatic pressure. So osmotic pressure is that pressure that is exerted on the membrane. Okay. Basically, you want to be able to get nutrients in and out, and it's all. Hopefully, you had some biology, and we talk about concentration of these concentration of and movement. From an area of higher concentration to an area of lower concentration, you can move from sugar across across a solution into a cell. By having the concentration of glucose higher outside the cell than inside the cell. So the pressure on that membrane by that solution will determine whether things can go across or not. Okay. Hopefully you've had this again in biology, but hypotonic solutions have a lower solute concentration. All right. Solvent is the stuff that it's dissolved in. Solute is what's being dissolved. So if we have a sugar solution, the water is the solvent and the sugar is the solute. So in a hypertonic solution, you would have, for example, more water than sugar. So you'd have this very low glucose solution. And if you have like an animal cell, you place it in a hypotonic solution, it ruptured, it bursts. This is why in a hospital, just FYI, we have to have normal saline, 0.9% salt with water. It's because if you you put an IV of water in there, it will rupture the blood cells. Okay, FYI. Hypertonic solutions are just the opposite. Okay, the solute concentration is big. And when cells are placed in these solutions, these cells will what we call crenate or pull in or shrivel up. All right, hopefully you've had this again in biology. This effect is what helps preserve foods when you put them in these salty solutions. Okay, the cells shrivel up and it makes it harder for them to spoil. We do have organisms that like salt, believe it or not. It does restrict the organisms to certain environments. We couldn't, live in a salt lake all day. If you go to the ocean, and you are in there for several hours and you get out of there, you notice your skin is kind of shriveling up. Yeah. Okay. So obligate halophiles, salt, can grow up into solutions of 30% salt. They love their salt. Facultative halophiles can tolerate these for a while. We have some like organisms that live in Great Salt Lake. They're perfectly fine. And that's what makes the color. All right. Hydrostatic pressure. This is water pressure. There are organisms that live in extreme pressure. These are barophiles, barometric pressure barophiles. Their membranes and enzymes are made to maintain their three-dimensional shape at those pressures. It's just like the organisms that live in the deep sea. You bring them up, they die because they are made to live in those extreme pressures. And their membranes and their enzymes depend on this to maintain the three-dimensional shape of the proteins and the membranes. But there are some organisms that actually require that high pressure. Okay. All right. So let's talk about how these guys associate with each other. I mean, we live with other species. We live with their own species. But we have relationships with these other species. And we may have an antagonistic relationship, a synergistic relationship, or a symbiotic relationship. And bacteria are the same way. Okay. So an antagonistic relationship is one where it's not a good one. Okay. You're an antagonist. It's bad. The micro harms. So if you get an infection into you, that microbe harms you. All right. So this is usually it benefits the aggressor. It uses up the nutrients. It uses up the physical space. They actively defend their environment. This is a real strong evolutionary driver, as I said, for maintaining these very complex ecosystems, even in the humans. Okay, aquatic biomes, that sort of thing. They keep populations in check. You have microbes in your gut. You have microbes in your mouth. You have microbes on your skin. As I said, the beneficial bacteria use this to prevent invading bacteria. So if you get your skin is basically, hopefully very patent, you don't have scratches and everything, and you get something drops on your skin that has bacteria, and it happens all the time. The bacteria in your skin will aggressively go after that bacteria and kill it. You've got a mouthful of bacteria, huge mouthful of bacteria. We call it normal flora. We'll talk about this because we'll look at this at the end of the course. But your normal flora is what prevents you from getting infections most of the time from whatever you're eating, because you can bring all. You're breathing in organisms, you're eating organisms. but your bacteria that are in your mouth are antagonistic to these organisms, and they fight them off. Okay, so and the same thing with your gut. You've got a lot of bacteria in that gut, and it really helps you fight these things off. Okay, so we want this certain times, we want this antagonistic relationship, and it's a positive one. Then we have a synergistic relationship when two or more organisms cooperate. Okay, um, and they can work together and mutually beneficial. And we'll talk about some of these, but sometimes you can have these synergistic relationships where an organism will produce something that the other organism needs. Okay, the microbes can survive independently, but they thrive better when they're together. Okay, maybe certain bacteria feed off of each other. All right. Then you can have a symbiotic relationship, and this is a close interaction between microbes and a host. And it does work with human health. We have three primary categories of symbiotic relationships: mutualism, commensalism, and parasitism. Okay, so mutualism, it's a mutual relationship. They both like each other. They get along. The host benefits. So I've got the example of the human gut biome. You've got trillions of bacteria live in your digestive tract, and they help break down these carbohydrates that you're eating, and they produce vitamins, and they help train the immune system. And, if they find something that's given to the immune system, and they get a steady supply of nutrients and a nice, warm, cozy place to live. So everybody's happy. Okay. Then you can have commensalism. This is where the microbe benefits and the host is neither helped or harmed. This staph F8, the human skin, they do consume the dead skin cells and the oils. It doesn't affect you. They can occasionally offer protection. As I said, they can aggressively go after certain pathogens that may be introduced on the skin. But we're not really the help is not real great like it is with mutualism, but they're perfectly fine and they don't hurt us unless, we were to cut ourselves out and drive this stuff into our skin. Parasites, the microbe benefits at the expense of us. And this is what happens with our pathogenic bacteria or viruses that cause us infection or disease. They are taking our resources away and we are getting sick. Okay. All right. The other way that these microbes like to live is called a biofilm. And this is a complex relationship among these microorganisms. And they develop this matrix so that they make a film and they adhere to each other. And it allows them to attach to a substrate or something like that, and sequester the nutrients, and they may protect the individuals in the biofilm, and they will form on a surface, often as a result of what we call quorum sensing. They will grow and grow and grow until they come to a point where they can't grow anymore and they stop. This is what happens, FYI, when we put a bacteria on an agar plate. If we spread it over there, it'll grow and grow and grow until it gets to the end, and it'll stop growing because it's sensing that that's the end. Some organisms can be more harmful when part of biofilm because it can be stronger, but at the same time, biofilms can be important. You form a biofilm every day. When you wake up in the morning, that film on your teeth, that's a biofilm that's formed from all the bacteria that are in your mouth. Okay. And then you brush it off. It's not, it's not a bad thing. It's protective, but, you can also, it's not something you want on there all the time. Okay. But it does form. That's an example of a biofilm. All right, so now that we've talked about the relationships and we've talked about the temperature and the pH and the oxygen requirements and all that, how do we grow these things and why do we grow these things? Okay, so we take this bacteria, which we call inoculum, and we introduce it into a growth media. It's either going to be a broth or a solid. And again, the broth or the solid is going to contain, hopefully, all the nutrients that organism needs to grow. And we have several different kinds of specimens. We can have environmental specimens. This is required if you work in a hospital and stuff like that. You have basically a department that makes sure they go around and test areas of the hospital every month. We used to, in the lab, we get environmental specimens from somewhere. You got to make sure if you're in a hospital setting, you got to make sure you don't have all these antibiotic resistant organisms growing everywhere, or if they are growing, you want to know where. So you go in and you test your OR, and you will test your call buttons, and you will test the buttons on the elevator, and you will test, for example, if you're doing physical therapy and you have these warm baths that people are putting limbs in, just for hot tubs, they'll test that, because those have to be cleaned between patients. And they will go in and they will culture. So these environmental cultures are done every month by law to make sure that, we don't have hospitals that are just overrun with organisms that can cause us diseases. Then we have the clinical specimens. These are the specimens that are collected on the patients and sent down to the lab for us to identify. And then we will store them. Okay, we store them for sometimes, it depends on the specimen we get, how long we can keep it. Okay, but usually culture will keep it, for 5 days, just in case. Culturing refers to the act of making these microorganisms grow and being able to work with them, just like you cultivate plants. Okay, same thing. Okay, well, how do we do that? Well, majority of prokaryotes have never been grown on a culture medium. If you think about it, the majority of prokaryotes that are out in the world have never been grown on a culture medium. So, just not there. But when you are working in a medical profession, we need to get a hold of these organisms that cause disease. We want to know what's causing the infection. Okay. And so we have 6 different types of media. You may come in contact with 2 of these. You won't probably come in contact with any of the other 4, unless you work in a lab or maybe a doctor's office or something like that. But the anaerobic media and the transport, you might come in contact with. So let's talk about these. All right. Supportive media or basal media, it supports the growth of most organisms. Now, these medias are what we call defined medias, generally because the chemical composition of them is defined. It's the same every time, okay? Sometimes when you do research, you're going to change these things, but ideally, you're going to have this. It's the same composition every time. We know what's in these augers, and they're going to be fine. So supportive auger, again, most of the other organisms will grow on these. You have to have something really weird if it's not going to grow on this. The most common is this tryptic soy, or we call TSA, tryptic soy agar, or tryptic soy broth, TSB. And it is organic extract, so you can see yeast and beef and some proteins. And the exact chemical makeup varies slightly per batch, but not enough to make any big difference. And it's ideal for growing these wide variety of these, what we call non-fastidious organisms. So like they're not picky. These guys are not picky eaters, is the bottom line. They you give them this, they're fine. We do have bacteria that are picky eaters. We'll get to that. All right, and there are some that like their growth media to be a little more nutritious, make a little more extra oomph in it. And so these are designed to grow some fastidious organisms that have some more complex nutritional needs. And the ones we use particularly are called blood auger plates. And this is just, it's all it is tryptic soy auger with a little bit of sheep's blood, 5% sheep's blood. That's all it is. It just likes that blood. This particular auger, and we'll talk about it a little bit later on, because you have another lab where I kind of repeat this and I show you the augers and everything, go into detail. But this allows us, some of these organisms have the ability to hemolyze the blood, and they have a certain pattern of hemolyzing. So we like to do these plates out on the blood, these organisms out on blood augers, so we can look at the hemolysis pattern and stuff like that. Blood auger plate is a very, very, very common auger to use. We use, and again, remember I said we don't use just one plate. We'll do more than one. So this and tryptic soy, those are very common ones to use. Charcoal it's a very interesting auger. It has charcoal in it, and specifically to grow out, believe it or not, pertussis. And it's, if you ever get it, if you're working in peas and you have somebody with pertussis, they'll ask for a pertussis culture, and the lab will say, I need charcoal auger, and they'll send it up to you, and because you got to have the charcoal, it needs that charcoal. So that's what I'm saying. It's picky. All right, we call it fastidious. So here's an example. I have the blood auger plate, and you can see these are hemolysis patterns. We'll talk about this, but this is beta hemolysis, alpha hemolysis, no hemolysis, or gamma hemolysis. And this puts it in a group. We use this particularly with streptococcus. It helps us decide which streptococcus is growing. This is charcoal auger. You can see the pertussis growing. You can see, yeah, that is a black charcoal-y auger. All right, then we have selective media. It selects for something, and this is helping you remember the names. So it contains a certain substance like a bile salt or a dye or antibiotic that inhibits the growth of certain microbes that we don't want to grow. Okay, so this helps us isolate these organisms. Most people don't understand, if you haven't worked in a lab, but you get a culture in and you have no idea what's going to be on that swab. We have no idea. I mean, this is coming from the gut, or this is coming from a wound. I mean, there are thousands, not thousands, but hundreds of organisms that can be growing from this wound. One of the quickest ways for us to help identify these organisms, and also to help us isolate the organisms so that we can quickly go on to the sensitivity and ID, because we have to have a pure culture of that organism in order to do the identification and sensitivity. We'll talk about that. And if I swab my throat, I've got all these organisms. Or if I get a urine in, I may have a couple different organisms in that urine. And so what we will do is we will try and separate these organisms a little bit to help us identify it. And what we can do is use these selective medias. McConkey, very often used, it selects for gram-negative. So any gram-positive organism that is growing will not grow on the McConkey. So if you've got a tryptic soy auger plate and a McConkey plate, and it's growing on the tryptic soy, but not growing on the McConkey, it's a gram-positive. Okay? Same thing. And if you've got mixed culture on one plate, but you got a pure culture of something on your McConkey, you know that McConkey organism is gram negative. EMB does the same thing. It has some dyes in it that will actually color, and we'll talk about that in a second. And then I have CNA that selects for gram positive. So we can actually, if we put it out on a couple different plates, we can right away separate our gram negatives from our gram positives, or hopefully get that done. Okay. Then we can use differential agar. I'm going to show you a picture of this. Contains some chemical indicators, some dyes that cause bacteria to look distinct. They change color. And it helps us distinguish them. So, for example, is EMB that we talked about before, this ESM methylene blue, it selects for gram negatives, but it's what we call a selective and differential plate because it selects for the gram negatives, but also helps us determine lactose fermenters from non-lactose fermenters by color. Okay, same thing with McConkey. It's a selective and differential. The blood agar plate I mentioned already. You can look at hemolytic versus non-hemolytic very, very quickly. It can, is this an alpha hemolytic organism or is it a beta hemolytic? Do I have a strep throat? That's a real easy way to pick out a strep throat, okay? And again, the CNA will do the same things. And so the agars can be selective and differential. So let's take a look at this. So here's these examples. This is the nutrient agars. This is TSA. You can see it's a nice, light yellow color. So you've got two organisms growing. So like this is, you get your plate, you look at your TSA plate, and this is what you see. But at the same time, you've plated out these organisms on McConkey or EMB. All right, well, I'm looking at it now. Look at the difference, okay? This is Staph aureus, which is gram positive, and E. coli, Escherichia coli, which is gram negative. And if you remember what we said, McConkey agar selects for gram negatives, okay? So on this plate, your gram positive, your Staph aureus is not growing. It's being suppressed. But your E. coli is doing great. Okay? Then in the McConkey agar, go back to my McConkey agar, and we said lactose fermenters versus non-lactose fermenters. Wow. Notice the color. Two different separate colors. So you could have two gram-negative organisms growing, and because you've got a differential agar that tells you whether it's a lactose fermenter or a non-lactose fermenter, you can separate it that way. So it's a way of us using these organisms and helping us determine what this organism is. All right, and then something you're probably going to get familiar with is transport media. Okay, this is a non-nutritive media, and basically what we're trying to do is keep the organisms alive after you've done a culture in a doctor's office or at a hospital floor. Keep it alive until it gets to the lab, and the lab can plate it out. Because I can tell you, when the cultures come down from the lab, most of the time they're not plated out immediately because we're busy down there, and we plate it out as soon as we can, but so you use a transport media or something. If you send us urine, we got the urine, but if you've got something where you've swabbed, you're using a dried cotton swab. That's not anything that's going to keep a microbe alive. So you've got to use a transport media to keep these guys alive. A lot of times you have a tube and you have to turn it upside down, break the little vial and make sure the liquid goes over the swab, but you got to make sure that those organisms stay alive. And particularly in the doctor's office, when the lab comes and picks up the lab specimens once a day. Okay, So transport media is exactly what it says. It's used to transport the organisms to the lab so that we can grow them out and keep them alive while they're in transport. Okay? Anaerobic cultures, the same thing. We've talked about anaerobic cultures saying, man, they can't get in touch with oxygen. They're special oxygen-free holding systems, and we use them they're used particularly in OR, because, I mean, an open wound is going to be, with an organism in it, it's going to be an aerobic bacteria. But when you get into the gut, that's an anaerobic environment. And so most of the time, these are coming down from the OR. And what they do is they'll swab that particular area, and then immediately goes into the anaerobic transport media. This is a gel. The end of the swab goes into the gel, and it prevents the oxygen from getting to that organism and killing it. Okay? So now that we've got it, we've got it collected and stuff like that. How does this grow? Okay, we're talking about growth. So we said each colony is basically, the same parent. So we divide by binary fission. So in about 20, 30 minutes, you will get 2 bacteria. Okay, you can see that little septum forming, and it's going to divide. In another 60 seconds, you got 4. It's a minute, sorry. And you can see in 120 minutes, you got quite a bit. Well, if it grows for hours and hours and hours and hours, you're going to have lots of bacteria in that single colony. Okay, that's how it does it. So that's binary fission. The generation time is the time that's required for that bacterial cell to grow and divide. And some go faster than others. And it depends on the conditions. If the conditions aren't particularly great, it's going to be slower. Okay. So that's why we need to make the chemical compensation and the physical conditions and stuff like that as ideal as it possibly can be for these guys. All right, in a closed system, which we have, like a culture, it typically follows what we call S-shaped curve that consists of 4 phases: lag phase, log phase, stationary phase, death phase. And this will track the population of the bacteria and their ability to multiply and divide and then eventually die. And I'll show you the curve in a second. But lag phase means you put that microbe in that culture material, and they got to adapt to their new environment. What in the world is this? They are metabolically active. They're synthesizing enzymes, but they're not yet dividing. They're not increasing in population. They're kind of getting ready to go. But they're getting used to their environment. Do I have enough nutrients? Do I have, you can't divide unless you got everything you need to divide. So it's kind of got, do I have everything to divide? Is the temperature good? Whatever. As long as that's good, you will fairly quickly go into log phase. And that is when these cells divide by binary fission at a constant rate. And again, the generation time is the time it takes for population to double, and the cells are dividing exponentially. Okay? And they just keep going. And then eventually, as they grow, as they are rapidly dividing, they are using nutrients. There's a limited source of nutrients in an agar plate. So these nutrients get depleted, and as you deplete these, toxic waste builds up. So the rate of the new cell division begins to slow down, and eventually you get a phase which is called stationary phase, where new cells are dividing about the same rate as the cells are dying. When you're in log phase, more cells are dividing than they are dying, okay? Quite a bit. But as you get towards the stationary phase, you get less of the cells dividing and more of them dying. So we start to get to the stationary phase. And then we get to the death phase, where the toxic conditions and the lack of resources are just we're not going to keep these organisms alive. So the number of dying cells exceeds the number of new cells dividing. And unless you enrich that agar with something, a broth, maybe add more broth, or take that culture and move it to another plate, that culture will die. Okay? And here's your curve. You have this short lag phase, and then this exponential log phase, and then the stationary phase and decline. Okay? And that's the way they grow. All right, so hopefully you've learned a little bit about microbial growth, and we will next talk about, I can't even remember what we're going to talk about. It's been one of these days. But after this, we'll get to talk about microbial control. And we've talked about them growing and what they need to grow. Well, we'll talk about how do we control them. All right, so we'll see you next time.